Bismillahirrahmanirrahim. Dear students, have a nice day. Hope you will be fine and best of your health. This is Dr. Sajid Mahmood Rao, Division of Science and Technology, University of Education, Lahore. Today, we are talking about phytochemical investigation on the constituents of Salvia centralinifolia and Indigofera heterantha. Salvia and Indigofera are the genus of family Labiatae and Leguminosae, respectively, which are the ornamental herbs and shrubs. We selected these medicinal plants because these are rich with phytoconstituents and have biological and enzymatic applications. So, in the context of these medicinal plants, we will discuss about isolation, separation, purification, and structure elucidations of the natural products. You can see over here the key contents of this topic, and these are isolation, extraction, purification, structural elucidations, Salvia centralinifolia and Indigofera heterantha. Dear students, in this class session you will be learned about how the organic compounds are separated, how the organic compounds are purified, and how the structural elucidation and determination is carried out of the natural products. Let's see what is isolation. Any of isolation work should be clarified in one's mind and should be concisely answered to the question. If asked, why you carry this isolation work? It is essential and core question. So you can answer the question. Purpose of isolation of natural product is broadly classified into one of the following three categories. The first is activity directing isolation. Second one, structure directing isolation. Third one is chemotaxonomical studies. So each of the above categories consist of different ideas and distinct isolation techniques. When we see the activity directing isolation, the work of this type is done only when the plant is known as bioactive, such as traditional medicines, or any activity under investigation is shown by the crude extract. The work is done to disclose what ingredients is responsible to the relevant activity. In structure directing isolation, you are searching new compounds or hopefully novel structure. Why did you choose this plant? Does it have possibility to yield novel compounds? You can confidently defend if asked why you choose this plant, even if it is not chosen with yourselves. Search literature as much as possible you can do. That is family, genus, species, local name and local uses of the plant. Search all references and compounds which has been isolated previously from the target plant or the structures known or unknown. Search literature for plants of the same genus of the similar uses. So, if there is some work on this plant, read that work carefully and critically. This literature survey will very much helpful to you. Otherwise, your work will be of little fruit because you start your work with poor knowledge. So, collect all information during the work. The more the knowledge is acquired, more it promises the fruits in the work. Once getting the sufficient information, then you start your work carefully. In chemotaxonomical studies, we subjected both the new and the unknown compounds for enzyme inhibition assay because the new compounds and the old compounds both are treated with equal weight in this category. So, bioassay using enzyme inhibiting activity is one of the ideal methods satisfying the above requirement. However, you always have to keep in mind that enzyme inhibitory assay is the assay of biochemical level and is not identical to biological or pharmacological phenomena of the whole body. Let's see what is extraction. Extraction is a process in which different phytoconstituents or the organic compounds are extracted by using different organic solvents, and that organic solvents are methanol, butanol, ethanol, ethyl acetate, chloroform and anhexane. So, extraction is the first step to separate the desired natural products from the raw materials. Extraction method includes solvent extraction, distillation method, pressing and sublimation according to the extraction principle. Solvent extraction is the most widely used method which consists of the following three steps. The first step is solvent penetrates into the solid matrix. Second, the solute dissolves in the solvent. Third, the solute is diffused out of the solid matrix. And the fourth is extracted solutes are collected. The extraction process is carried out smoothly by using the following factors: the properties of the extraction solvent, the particle size of the raw materials, the solvent to solid ratio, the extraction temperature, and the extraction duration will affect the extraction efficiency. Now you can see over here the difference between the extraction and isolation. Researchers have used these terms interchangeably while describing procedures of extraction or isolation of essential oils from aromatic plants and extracts of the medicinal plants. However, extraction is a process used to obtain compounds. Compounds mean large number of compounds, bunch of compounds, which may be unrelated from plants' materials, stem, bark, leaves, and roots, while isolation is a process of separations to obtain a pure single compound from the plant extract. Purification of organic compounds is carried out by different techniques. However, we use general chromatographic techniques for the isolation and purification of the natural products. Almost everything that we see these days impure, isn't it? The water we drink and the food we eat also need to go through the levels of purification processes. Similar is the case with the organic compounds. There are several methods of purification of organic compounds. Why are these important and how do we do it? Let's see what is the types of purifications. A large number of methods are available for the purification of substances. The choice of the method, however, depends upon the natures of the substances, whether solid or liquid. It also depends on the type of impurities present in it. We commonly use these methods for purification of organic substances. Simple crystallization, fractional crystallization, sublimation, simple distillation, fractional distillation, distillation under reduced pressure, steam distillation, azeotropic distillations, and chromatographic techniques. Let's see what is chromatography. Chromatography works on the principle that different compounds will have different solubilities and adsorptions to the two phases between which they are to be partitioned. Thin layer chromatography is a solid liquid technique in which the two phases are a solid stationary phase and a liquid a moving phase. So TLC is the single technique which is used for the purification and for the conformations of the organic compounds. As you know, if we have a single component on the TLC cards, it means the compound has been pured. So thin layer chromatography is a technique which is used manually for the conformations and the purifications of the organic molecules. Let's see the general overview of this topic. As you know, God has gifted Pakistan with enormous varieties of medicinal plants. Out of these, only 20% have so far been carried out for their therapeutic effects. So, there is a need of well-organized and coordinated work in the field of natural product chemistry. Today, we will discuss about the isolation and purification of natural products in the context of pharmacochemical studies on two indigenous medicinal plants of Pakistan, namely Salvia centralinifolia and Indigofera hatrantha. Salvia centralinifolia is the largest genus of aromatic and ornamental herbs and shrubs. Belongs to family Abiatae, which comprises of 210 genera and nearly 3,500 species, mainly occurring in Pakistan, India, and China. When we see its pharmaceutical importance, the phytoconstituents which are present in this genus are used as folk medicines, treatment of coronary heart diseases, particularly in angina pectoris, myocardial infarction, amenorrhea and dysmenorrhea, and insomnia. A large number of compounds of this genus have antiseptic, carminative, diuretic, spasmolytic, and hemostatic activities. Seeds and roots of this genus are used for cough, hemorrhoids, chronic infection of skin, heel wounds, anti-diabetic, appendicitis, throat complaints, and digestive tract system. The topic is comprised of two phases. In the phase 1, that is called phytochemical screening and biological activities of the plants. This phase consists of the following steps. The first step is plant collection. Plant collection is carried out on the basis of literature survey as well as the biological and the enzymatic activities of the plant. After that, plant is submitted to the botanist, which confirm its genus and species and allow us to proceed it for further process. Next step is shade drying. At this step, the whole plant material is placed in the shade for drying purpose. This is carried out within 7 to 15 days. After that, the whole plant material is chopped and grinded by using pestle mortar or by using crusher and grinder if the quantity of the plant material is huge. After that, the powder material is dipped in methanol. Here it takes 20 to 30 days if the plant material is large quantity and a few hours if the plant quantity is less. Then the methanolic extract is filtered and we get two parts. One is residual part and the other is filtrate. At this stage, the residual part is discarded and the filtrate is subjected for further process. The filtrate part is now called the crude methanolic extract. This methanolic extract now is subjected for fractionation. Fractionation is carried out by using different organic solvents from less to high polars that include hexane, chloroform, ethyl acetate, methanol, and butanol. So we get 4 to 5 different fractions on the basis of the Pilates system. After that, each fraction is subjected for phytochemical screening. In the phytochemical screening, we get which fraction consists of maximum and minimum number of the phytoconstituents and which fraction is more active phytoconstituents. So, on the basis of phytochemical screening, the active fraction is subjected for further process that is called in vitro study. The second phase consists of isolation, purification, and structural elucidation of the natural products. This phase consists of the following steps. In the first step, the more active fraction is chosen on the basis of phytoconstituents and maximum number of organic compounds for the column chromatography. So we choose a chloroform fraction which is subjected for column chromatography for the separations of the organic compounds. Column chromatography is a technique which is carried out by using stationary phase and the mobile phase, and after the elution of the column we get the different kinds of organic compounds that are in the form of separation on the basis of Pilates system. The separated compounds are then submitted for thin layer chromatography which confirm its purity. TLC or thin layer chromatography is a technique which is used to confirm the purity of the organic compounds. If we have a single compound or component on the TLC card, it means the compound has been pure. When the confirmation is confirmed, then the pure compounds are submitted to spectral techniques. Spectral techniques is a tool which is useful to elucidate or determine the structures of the known or the unknown compounds. Spectral techniques consist of C13 NMR, proton NMR, UV, IR, HMBC, HMQC, NOI, COSY, and MALDI techniques. So, when the spectral techniques is completed, after that we get a complete file and we submitted this file for the spectral solvation. So, after the solvation of the spectra, we get the known or the unknown compounds. For example, we have two known compounds, and these are confirmed on the basis of the literatures. Means comparisons of spectral data with the literature confirm the identity of both the compounds 1 and 2. Let's see Which kind of the phytoconstituents are present within the genus Salvia centralinifolia? Literature survey shows that the members of this genus are rich with essential oils, mono and sesquiterpenes, triterpenes, steroids, flavones, xanthones, lignans and neolignans, and pigments. The medicinal importance and biological activities of genus Salvia prompted us to investigate one of its species, namely Salvia centralinifolia, on which no work phytochemical has so far been carried out. So, the extract was subsequently divided into an hexane, chloroform, ethyl acetate, N-butanol, and aqueous fraction. The present investigations on the chloroform soluble fraction resulted in the isolation of 3 new compounds along with 12 known compounds reported for the first time from this species. The structures of these compounds were established through spectroscopic techniques including 1D and 2D NMR experiments. These compounds have been reported and isolated from Salvia centralinifolia. All these molecules are lignans. Lignans are those organic phytoconstituents which have been isolated from wood of the plants. The lignans consist of phenyl-propanol units, and this unit linked with each other at carbon 7 and 9 position to make the lignan molecules. Let's see all these lignan molecules, their sources, their uses, and their solubilities. The first compound is fluoxyresinol. This is isolated in amorphous powder. This molecule consists of different methoxy and hydroxyl group which are linked with the phenyl molecules, and phenyl molecules are linked with each other by phenylpropanoid units and the tetrahydrofuranine moiety. This molecule is isolated and purified from the roots of Ilex pubescens, and it is soluble in chloroform, dichloromethane, ethyl acetate, dimethyl sulfur oxide, and acetones. Fluoxyresinol is used as antibiotic, anticonvulsant, antioxidant, anti-inflammatory, immunomodulatory. It is used as analgesic. It can also be used as antimicrobial, antiviral, and antihypertensive agent. Fluoxyresinol is used as anti-cancer, anti-hyperglycemic, anti-convulsant, and wound healing activities. The second molecule is lariceresinol. This molecule is isolated as colorless needles. It also consists of 2 phenyl units in which methoxy and hydroxyl group are attached. In this molecule, tetrahydrofuranine moiety is linked between the 2 phenyl units. So, lariceresinol is found in sesame seeds and in brassica vegetables. It is also found in the bark and wood of white fir. Lariceresinol is soluble in dichloromethane, chloroform, and DMSO. When we see its uses, it inhibits the proliferation of cancer cells, suppress the flu virus, and work as antimicrobial activity. This molecule provides therapeutic effect to cardiovascular tissues by promoting vasodilation and reducing fibrosis. It is also used in inflammation, apoptosis, and oxidative stress. This molecule have neuroprotective effect as well as general antioxidant and anti-inflammatory effects. So it is also used to prevent angiogenesis. Taxiresinol is isolated as a colorless needles. It is similar to that of lariceresinol. However, there is a difference only due to the presence of the methoxy and the hydroxyl group at different positions of the carbon atoms of the phenyl units. Taxiresinol is isolated from the branches of Taxus valencina. It is soluble in DMSO acetone ethyl acetate. Taxiresinol shows anti-cancer and anti-allergic activities. It shows inhibitory activity on induced histamine release from the human basophilic cell lines. Phenolic lignan is isolated as white flax. It is isolated from soybeans, cruciferous vegetables, and such as broccoli and cabbage. Phenolic glycerine is soluble in ethanol, methanol, DMSO, phenol, and dioxane. When we see its uses, it is used as antioxidant and free radical scavengers, leading to decreased risk of cancer development in the human body. Next molecule is diacylglycinol, which is isolated as amorphous solid. Diacylglycinol is isolated from Plichia indica, and it is soluble in ethanol, acetone, acetic acid, and peracetic acid. It is used as anti-inflammatory, expectorant, tranquilizer, antispasmodic, anticonvulsant, antifungal, and antipyretic agents. Next molecule is 3-beta 6-alpha dihydroxylupiene 2029-ene. This molecule is isolated from the less to medium polar fraction of the plant. This is a terpenoid molecules. Terpenes are those molecules which consist of isoprene units, and the isoprene is C5H8 unit, which constitute the basic building block of the terpene molecules. So this molecule is isolated from different genus of the Salvia, and it is soluble in DMSO. It is soluble in dimethylforamide, methanol, and ethanol. This molecule works as antimicrobial activity and anti-inflammatory agents. Erythrodiol is a triterpene molecule which is isolated as amorphous powder. This molecule is isolated from the olive plants, and it is soluble in benzene, ether, dichloromethane, and DMF, that is dimethylforamide. This molecule is used to decrease the level of antibodies and blood-brain barrier leakage and infiltrations of inflammatory cells within the central nervous system. Dilenic acid is also a tetraterpene molecule which is generally isolated from different plants from the less to high polar fractions of the plants, and it is isolated as a amorphous solid. The source of this plant is Dilenia pepuna. It is soluble in toluene, dimethylforamide, dichloromethane, ethyl acetate, and dimethyl sulfur oxide. This molecule is used as antibacterial, antimicrobial, and cytotoxic agent. Next molecule is lupiol. This molecule is isolated as colored as needles, and it is also known as mangoterpenes. Lupiol occurs in the skin of lupine seeds as well as in the latex of fig trees and of rubber plants. It is also found in many edible fruits and vegetables, white cabbage, pepper, cucumber, tomato, in fruits such as olive, fig, mango, strawberries, red grapes, and in medicinal plants such as American ginseng and sea butter plant. This molecule is isolated from lupiol organic solvents such as ethanol, dimethylforamide, which should be purged with an inert gas. This molecule is used as anti-inflammatory, antimicrobial, antiprotozoal, anti-proliferative, anti-invasive, anti-angiogenic, and cholesterol-lowering agent. Beta-amylin is isolated as colorless needles after washing with ethanol. This molecule is isolated from Salvia centralinifolia, as well as this molecule is isolated from the stem bark of Estonia. Beta-amylin can be found in a number of food items such as thistle plant, pepper, and endive plants. This molecule is soluble in acetonitrile, tetrahydrofuran, and ethanol, and it can be used as exhibits long-lasting anti-nociceptive and anti-inflammatory properties in two models of persistent nociceptions via activations of the cannabinoid receptors. Oleanolic acid is isolated as colorless needles from this plant. This is the tetraterpene molecule and is isolated from the less to medium polar fractions of the plant. Oleanolic acid is isolated from the fruits of Olea europaea, that is olive tree. This molecule is soluble in carbon tetrachloride, dimethyl sulfur oxide, dimethylforamide. Oleanolic acid is used in Chinese medicine for the treatment of liver disorders such as viral hepatitis. The next molecule is beta-cetosterol glycosides. Beta-cetosterol glycoside is isolated as amorphous solid from this plant, and this is a steroidal molecule in which glucose moiety is attached at 1 to 3 positions of the carbon atoms of the steroidal molecules. This is also isolated from the less to medium polar fractions of the plant and found in rice bran, wheat germ, peanuts, corn oils, and soya beans. Beta-cetosterol is soluble in acetic acid, chloroform, acetone, and acetonitrile. It reduces cholesterol and helps in cure of inflammatory diseases. These are the new molecules or the organic compounds which have been isolated and extracted from genus Salvia and species Centralinifolia. So these molecules are tetraterpenes or terpenoids which have been published in Archives of Pharmaca Research in 2006. The first two molecules, Salvin A and Salvin B, are the terpenoids, and the third one is lignin molecules. The lignin molecules named as centrolinol, which have been published in College Journal of Chemistry in 2008. Let's see how these molecules have been separated, how these molecules have been purified, and how the structure of these molecules have been developed by using different spectral techniques. Let's see how the extraction and fractionation of Salvia centralinifolia is carried out. The whole plant material, that is 14 kg, is ground, powdered and extracted with methanol and concentrated on rotary evaporator. So we obtain methanolic extract, that is 550 grams. The methanolic extract is partitioned between N-hexane and water, and in this way we obtain further two fractions, that is aqueous fraction and hexane soluble fraction. The aqueous fraction is further extracted with chloroform and ethyl acetate respectively, and we obtain further two more fractions, that is chloroform soluble fraction and ethyl acetate soluble fraction. The aqueous fraction at the end is further extracted with butanol, and we obtain two fractions, that is butanol fractions and aqueous fraction. In this way we obtain total 5 fractions on the basis of organic protein system, that is in the increasing order of the organic solvents. That fractions are hexane soluble fraction, chloroform soluble fraction, ethyl acetate soluble fraction, butanol fractions, and aqueous fraction. Now we have to decide which fraction should be subjected for the column chromatography on the column silica. So we choose here the chloroform soluble fraction for further column chromatography to obtain and separate the organic compounds. The chloroform soluble fraction, that is 65 gram, is subjected to column chromatography over silica gel using an hexane chloroform, pure chloroform, chloroform methanol, and methanol 100% in the increasing order of purity. In this way we obtain 4 subfractions, and that fractions are fraction Ca, fraction Cb, fraction Cc, and fraction Cd. The fraction Ca is further subjected to column chromatography over silica gel by using the organic solvent purity system is 9.8 and 0.2% hexane chloroform. So we obtain two compounds, and that compounds are 13 and 15. The conformations of the purity of these compounds is checked by thin layer chromatography, and the organic solvent system which is used to confirm the purity or the purity of these compounds is 9.5 and 0.5% hexane chloroform for compound number 13 and 9.4 and 0.6% hexane chloroform for compound number 15. The fraction Cb is further subjected to column chromatography over silica gel eluting with 9.3 and 0.7% hexane chloroform. So it shows two major spots on TLC, and we obtain two pure compounds, and that compounds are compound number 9 and 14. The purity of these compounds were confirmed by using the organic purity system, that is 9.7 and 0.3% chloroform methanol for compound number 9 and 9.5 and 0.5% chloroform methanol for compound number 14. The fraction Cc is subjected to column chromatography over silica gel, and eluting with 100% chloroform we obtained compound number 3. The conformations of the purity of this compound is confirmed by using organic purity system, that is 9.9 and 0.1% chloroformethanol, which is confirmed the purity of this compound. The fraction CD is subjected to column chromatography over silica gel and eluting this column by using purity system 9.9 and 0.1% chloroformethanol, and we obtain the final pure compound, that is compound number 1, and we used the purity organic purity system for this compound is 9.8 and 0.1% chloroformethanol. So in this way, different kinds of compounds have been paired by using column chromatography, and their confirmations of the purity has been confirmed by different organic purity systems for the purity of the organic compounds. Chloroform soluble fraction, that is 65 gram, is further subjected to column chromatography over silica gel by using organic purity system is N-hexane chloroform, that is 0 to 100%, and chloroformethanol, that is 0 to 100%. So in this way we obtain 4 further subfractions, and these fractions are fraction CE, fraction CF, fraction CG, and fraction CH. The fraction CE is subjected to column chromatography over silica gel eluting with 9.8 and 0.2% chloroformethanol, and it shows 3 single spots on TLC cards individually, and we obtain 3 compounds: compound number 10, compound number 11, and compound number 12. The compound number 10 is confirmed its purity by using organic purity system, that is 9.6 to 0.4% chloroformethanol, and compound number 11 is confirmed by using 9.3 and 0.7% chloroformethanol organic purity system, and compound number 12 is confirmed by using organic purity system 9.5 and 0.5% chloroformethanol to check the purity of this compound. The fraction CF is subjected to column chromatography over silica gel and eluting with 9.7 and 0.3% chloroformethanol, we obtain compound number 2. The purification of this compound is confirmed by using organic purity system, that is 9.6 and 0.4% chloroformethanol. The fraction Cg is subjected to column chromatography over silica gel eluting with 9.6 and 0.4% chloroformethanol. In this way we obtain a mixture of 2 to 3 compounds. That mixture is further subjected on column chromatography over silica gel and eluting with 9.3% and 0.7% chloroformethanol. In this way we obtain 2 compounds, the compound number 4 and 6. The purity of these compounds was confirmed by using the purity system is 8.5%, 1.2%, and 0.3% chloroform, methanol, and water respectively. So in this way final purification by preparative TLC is confirmed by using this organic purity system for the compound number 4 and compound number 8. The fraction CH is subjected to column chromatography over silica gel and eluting with 9.5 and 0.5% chloroform methanol. In this way we obtain 3 compounds which shows a single spot on thin layer chromatography cards, and these compounds are compound number 5, compound number 6, and compound number 7. The compound number 5 is confirmed its polarity and purity by using 9.1 and 0.9% chloroform methanol, while compound number 6 is confirmed its purity by using 9.0 and 1.0% chloroform methanol, and finally the compound number 7 is confirmed by its purity by using 8.8 and 1.2% chloroform methanol. So in this way different kinds of organic compounds have been paired by using column chromatography as well as thin layer chromatography techniques. So after the purification of compounds, these compounds are submitted for the structural elucidations and determinations of the organic compounds for the spectral techniques. And these organic spectral techniques are IR, UV, proton NMR, C13 NMR, HMBC, HMQC, NOE, COSY, and MALDI techniques. So the structure of compound 1 is confirmed on the basis of these techniques, and the name of this compound is Salvin A. This structure has been confirmed on the basis of all these spectral techniques. The name of this compound is 2-alpha 3-beta 5-alpha trihydroxy or seen 12-in-20-ic acid. Now we discuss the physical parameters of these compounds. This compound is isolated as colorless amorphous solid and the amount of this compound is 15 mg. The alpha D of this compound is -74.40, that is in which carbon is equal to 0.062 in methanol. The IR is taken in potassium bromide disc and the lambda max of this compound is 3444 1700 and 1650 per centimeter. The HR EIMS mass over charge ratio, that is 488.3496, that is the molecular formula and molecular mass of these compounds. The calculated molecular formula of this compound is C30H48O5, that is equal to 488.3501. Further, the other discussions and the confirmations of this molecular structure will be confirmed on the basis of spectral techniques that has been discussed in the next slides.